Hello everyone, this is Brinda, Research Scholar, Department of Bioinformatics, Pondicherry University. I'm going to present my work on microbiome of 4 different lung diseases, a comparative 16S rRNA metagenomics to understand the diversity and functional profile. So in this study, we collected 4 different lung diseases from NCBI and it's subjected to taxonomy assignment followed by functional analysis. So firstly, it was aimed to compare the microbiome of all the lung disease sample with a healthy sample and to show the effect of cigarette smoking in male and female patients by evaluating the abundance and diversity across the sample. Second is that finding the possible biomarker to treat or monitor the disease severity and co-infections. Finally, profiling the functional properties associated with the disease lung microbiome. Then in this study, we collected, as I told before, 4 different lung diseases. We were having some set of criteria to collect the sample. So first is that sample with lung diseases. So it should be collected from respiratory tract and sample with gender and smoking history, and the source would be 16S metagenomic and paired in. So with this criteria, we collected pneumonia, associated HIV, lung cancer, sarcoidosis, and COPD. Then after collecting the sample, we went for pre-processing by using the Galaxy FastQC and Trigonometric tool. And also we use SLIVAngs, the inbuilt Silva incremental aligner. Then after pre-processing, that filtered sequences were subjected to dereplication. The dereplicated sequences were mapped against the Silva database with a similarity of 93%. Then that samples were classified and clustered together and assigned to OTU table. Then that OTU table was consolidated from all the microbiome, then normalized using TSS normalization method. That normalized table was subjected to diversity, statistical and functional analysis. So in the pre-processing section, we can see the number of samples with respect to male and female and smoking history. Then finally, that control sample also. Here, except pneumonia, remaining all the microbiome were having the control. So the pooled control sample from these 3 were the control for pneumonia. Then here we can see the sequence quality also. All the samples were having the Phred score of more than 20. So here I have given that example quality check plot. Then this plot is called rarefraction plot. It will tell you that sequencing depth and also OTU abundance. Then in result section, we started with disease lung microbiome diversity and abundance. For diversity measurement, we used PAS4, the standalone tool. It is having the set of parameter to measure the diversity of the lung microbiome. So among these 4 microbiome, which one is the most diversified? That is the aim of this particular task. So we found like lung cancer is the most diversified among all based on this ecological parameter. Then the relative abundance of different lung disease microbiome from phyla to family as listed here. So here at phyla level, we found like firmicutes, actinobacteria, bacteroides, and then finally proteobacteria are the topmost abundant phyla we found in this study. And after checking the literature, we found like these are the common phyla which will present in the microbiome of human. Then for a genus level, we use that Venn diagram representation. From this plot, we can tell like what are the common as well as the what are the unique genus for each lung microbiome. So we found like 211 genus were common to all the lung diseases, including escape pathogen. So specifically for escape pathogen, to compare against the control, we use star bar plot. In that, we found like these escape pathogens were common to all the lung diseases, and these are abundantly seen in almost all that microbiome. Then to check the probiotic level, um, compared to control sample, the probiotics, bifidobacterium, lactobacillus were comparatively less in comparatively less in disease lung microbiome compared to lung control samples. Then we went for network analysis also using past correlation, compositional data, network analysis. We found like Haemophilus, the respiratory pathogen, which is a common hub in all the lung diseases with the smoking history. And in non-smoker, we found like Alloprevitella, the moderate pathogen found in all the lung disease sample. And then along with that Haemophilus, we found escape pathogens such as Acinetobacter, Enterococcus and Klebsiella are also the hub in all the lung diseases with smoking history. Then with respect to gender, we found except COPD, remaining all three lung diseases were having a significant difference among smoker and non-smoker. But in COPD, we found like non-smoker is the most significant difference. Then we went for functional analysis. For functional analysis, we used Megan, that standalone tool. In this analysis, we found like membrane transporter is the common and enriched function in all the lung diseases compared to control sample. The 5 to 15-fold higher in all the lung diseases. Then we went for CAT database analysis. In this CAT database is the comprehensive antibiotic resistant database. So in this database, we found like tetracycline resistant genes were high in all the lung diseases compared to this control. Then we went for pathway analysis using CATCH. So here we found like beta-lactam resistant pathway is enriched in that all the lung diseases, and the resistance is mainly due to RNA transporter. So RNA transporter are the transporter which present in the bacterial membrane. So the mechanism of RNA transporter is that effluxing the antimicrobial agent from inside the cell to outside the cell. So blocking this or targeting this membrane transporter from all the escape pathogen found in this study is one of the way to reduce the disease severity and co-infection of the lung diseases. And along with that, supplementation with probiotic also one of the way to reduce the severity and co-infections. So these are the references I gone through in this study. Thank you.